Okay, so yeah, I will get started, and in case you are not able to finish, you can do it later after I am done. Um, we discussed about pairwise alignment, and we were also discussing about multiple sequence alignment. But now I will get in more detail. So multiple sequence alignment means alignment of 3 or more proteins or DNA sequences. And when you visualize multiple sequence alignment, you are visualizing the conservation at different columns in the multiple alignment. So the aligned residues in a column uh, are homologous in structural sense, or could be homologous, of course, in evolutionary sense, could be in functional sense. Okay, they could be conferring similar or related functions. So let's consider multiple alignment of 5 globin proteins, and we will use several programs: Cluster W, Praline, Muscle, Propcons, and T-Coffee. We will focus on a histidine residue, H residue in the multiple alignment, because it has a critical role in binding oxygen. So you would expect that histidine is conserved, so it should be aligned by these programs. So let's see actually cluster W and focus on histidine, which are indicated by inverted triangles. So one is open and the other is filled. Okay. So don't worry about this phenylalanine. Just focus on histidine alignment. So you can see at this position, it is histidine is aligned very well in all these 5 globin sequences, and they are from beta globin, myoglobin, neuroglobin, soybean globin, and rice globin. But if you look at the other histidine, it seems like it got aligned in beta globin and myoglobin, but it is not so with neuroglobin, soybean globin, and rice globin. So it is offset by one residue, right? It is not aligned. All these histidine residues are not aligned in one column. Can you see that? So look at this, histidine, but then this is offset by one residue, right? This is in the next column, and then this is also next column. So they all are not aligned in one column. Let's look at praline. So keep the mean focused on this. So again, this is also not good. So histidine in first 2 sequences are aligned, but then they are offset by several residues, okay, in the other sequences. Muscle is also not able to align histidine in one column. Propcons, and this is based on hidden Markov model, probabilistic model it uses, and you can see that histidine is aligned in one column in all these sequences, okay. T-coffee, this is also good. All histidines are aligned in one column, okay. So this way actually you assess the performance of these programs. If you know that these residues are important, they should be aligned, so it helps you in evaluating which program is the best in performing multiple alignment. But remember, uh, what you observe here, it doesn't mean that PropCons and T-Coffee are the best programs. This is just based on one test. You will have to do many tests in order to conclude which program is the best program. Okay. Okay. So multiple sequence alignment properties, and I will go to the last actually. So for 2 proteins sharing 30% amino acid identity, so 2 proteins with only 30% amino acid identity. So it is close to the twilight zone. You remember twilight zone? So what is the threshold for twilight zone? Yeah, but what is percent identity? 20, yeah, 20. So 20 and below actually twilight zone, and it is getting close to twilight zone. So for two proteins sharing 30% amino acid identity, about 50% of the residues would be superimposable in their structures. Okay, what this indicates? So at sequence level 30% identity, at a structure level 50% identity. Okay, what this indicates? Protein sequence 30% identity, structures 50% identity. What it means? The amino acid mutated in such a way that their results can be unusual. Structure may be same, but like aromatic will convert to another aromatic. Positive amino acids convert to positive amino acids. Okay, so physicochemical properties, but structures are more complex that way. If you think of properties, other properties also, because structures are three-dimensional conformations. They are complex. Other properties also you would want to consider, and then when you superimpose, then superimposition could be because of all these different structural properties, including physicochemical properties. Okay, so 30% sequence identity, 50% structure identity, which would indicate that structures evolve slower than sequences, right? So this is the conclusion that you will draw. Okay, so some aligned residues such as cysteines that form disulfide bridges, they are highly conserved because there is a functional constraint that cysteine should not be mutated, otherwise the function will be disrupted, right? Because of the functional constraint, cysteines could be conserved, highly conserved, and they should appear in a column highly or completely conserved in the proteins. So let's see, we will see that. There may be, so features of multiple sequence alignment, there may be conserved motifs, for example, in transmembrane proteins. So you can see domains in multiple alignment, okay, regions that are conserved across many sequences of a protein family. There may be conserved secondary structure features, and also when you look at multiple alignment, you will see consistent patterns of insertions or deletions. So gaps, they appear together often. Okay, so ungapped regions, then gaps, and then ungapped regions, and then gaps, right? So consistent patterns of gaps. So these are the features of multiple alignment. So we will see actually multiple sequence alignment is more sensitive than pairwise alignment in detecting homologs. Why it is so? Multiple sequence alignment is more sensitive than pairwise alignment in detecting homologs. Why it is so? Okay, very good. Anything else you want to add? Which one? Input is more. Okay. Anything else? So pairwise alignment is alignment of only two sequences, right? And we saw the challenges in aligning sequences like beta globin and myoglobin. They both belong to globin family. When you did, then BLAST says no significant similarity found. Okay, and for that reason, actually, BLAST was developed, and BLAST performs multiple alignment. You remember, and multiple alignment helps in uh, coding or whatever you say, encoding the patterns that are representing a protein family. Okay, peculiarities or characteristics of protein family are captured in a multiple sequence alignment. Okay, and because of that, multiple sequence alignment becomes more sensitive in identifying members of a protein family or gene family. Clear? Because each family has some characteristics and they are coded in their sequences. When you include all sequences, then you can see those patterns, those conserved patterns that are characteristics of protein families. Okay, so if you are able to decipher those patterns, then you should be able to, through multiple alignment, then you should be able to identify more members of that family. Okay, such patterns may not be easily captured using pairwise alignment, because pairwise alignment generally only two sequences. Okay, a single query can be searched against a database of multiple sequence alignment, and we discussed about RPS BLAST, reverse position specific BLAST. Do you remember what it is? Reverse position specific BLAST. So what the database consists of? Okay, in the BLAST discussion, we learned about PSSM, position specific scoring matrix, right? And position specific scoring matrix is derived from multiple alignment. So begin with a query sequence and then BLAST, obtain their hits, perform multiple alignment, and thus you get the model which is representing the matrix, right, which is representing the multiple alignment. So that model or matrix, I think I had shown you, is position specific scoring matrix. Okay, that represents a protein family. So in the RPS BLAST, you have a database of position specific scoring matrices representing different protein families. Okay, and then you begin with a query sequence and BLAST against a database of position specific scoring matrices, which are models representing different families. Okay, so the model that best matches or fits with the query sequence is the one that the query sequence belongs to. Okay, model represents a protein family. So the model that is the best match with the query sequence is the one to which the query sequence belongs to. So basically the protein family that is represented by that model. Clear? Okay, so a single query can be searched against a database of multiple sequence alignments, and that could also be in PFAM database. Regulatory regions of genes may have consensus sequence identifiable by multiple sequence alignment. So regulatory sequences, promoter sequences. For example, transcription factor binding sites, where transcription factors bind. Okay, and they bind, which means that there is some function associated with those non-coding sequences, so they should be conserved, right? To identify those conserved patterns, you would want to perform alignment of those regulatory sequences and thus find consensus sequences, which means conserved sequences, conserved nucleotides in the promoter sequences, okay, in the transcription factor binding sites. And that can be deciphered using multiple sequence alignment, those conserved patterns. Okay, so how to score multiple alignment? Okay, so we saw and we discussed about scoring pairwise alignment. So given pairwise alignment, the score is the sum of the scores for alignment of residues at each position in the alignment, right? So you get a score for first position, you get a score for first position alignment of residues, and then second position, third position, you add them up, and that is the score for the alignment of two sequences. Okay. So for this, again using the same logic, you can say that you can get the alignment for alignment score or a score for each of the columns, and then sum over all the columns, and that would be the score for the multiple sequence alignment. Okay. So M is multiple sequence alignment and I is ith column. So you get a score for each of the columns, and then sum over all the columns, you will get the score for the alignment. Clear? Okay. So basically, if you have the multiple alignment, then you have to compute the score for each column and then sum those up, and that would be the score for the alignment. So let's see, actually there are many ways to do this, and let's see how we can compute these scores. And one could be the probabilistic scores. Okay, the probability of a column, assuming that each of the nucleotides or residues in a column occur independent of each other. Okay, so for example, if you consider this column has residues G, K, R, A, R. Okay, so what is the probability of R in this column? No, so I am looking at the last column actually in first row. So last column, this one. So okay, I am saying that this is row 1, row 2, row 3. Okay, okay. So 2 by 5, right? 2 by 5. So you can compute the probabilities for the residues, and then assuming that each residue occurs independent of the other, so you can compute the probability of this column. Okay, so probability of this column is probability of G multiplied by probability of K multiplied by probability of R multiplied by probability of A multiplied by probability of R. So there are two R's, so that would be probability of R square, right? So which is shown here, probability of R and count 2, right? And product, which means that you have to consider all amino acids for that column. Okay, and this is the probability of the probability of the ith column of the multiple alignment M. Okay, so you can get this. This is the score for the ith column of multiple alignment M. And take the logarithm of this. So take log. So if you will take log, then the product becomes sum. Okay, so take the log of this. So this becomes sum, and then this count. So log of this, so count logarithm p. Okay, and then this becomes sum. Okay, so let's look at a column, and where is that? This one. Okay, star here. So P, 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 P. So 100% conserved, right? So what is the probability of P? 1. Okay, 1. So log 1 is 0. 0. So imagine that each column is completely conserved. So what would be the score for the multiple alignment? 0, because basically you are summing up the scores of the columns to get the score for the alignment, right? Okay, so and this is akin to entropy. So if it is 100% conservation, then it is completely ordered system. Entropy is 0. Okay, as entropy increases, it indicates there is more disorder, right? Here it indicates that the multiple alignment has more changes or mutations, more variabilities are there in the multiple alignment. Okay, more conservation, less score. Okay, 0 is the best, and as it increases, it indicates that there are more substitutions in the multiple alignment. Okay, so this is one metric for computing the score of the alignment. Got it? The other approach is called sum of pairs approach, and this is also a simple approach where what you do is you consider all pairs for each column, okay, and look for their scores in the blossom matrix. So again, let's consider this column and all pairs. So G aligned to K, G aligned to R, G aligned to A, G aligned to R, okay, and then K aligned to R, K aligned to A, K aligned to R, R aligned to A, R aligned to R, A aligned to all pairs, okay. And then for each of these pairs, you are going to look for the score in the blossom matrix. Get the scores, sum them up, okay. And then you can do for the other columns, add all those scores that you will get from the blossom matrix, add them up, and that would be the score for the multiple alignment, okay. Okay, so this is called sum of pairs scoring. Now methods. Just like pairwise alignment, where you can use dynamic programming algorithms for obtaining the optimal alignment, you can use the dynamic programming algorithms for multiple alignment also. But it becomes really very complex. Computationally, it is just not feasible to obtain the best alignment using this method. So heuristic methods have been developed for multiple alignment, and one of such methods is called progressive methods, where a guide tree is constructed, which is similar to a phylogenetic tree, and then that guide tree guides how these sequences have to be aligned one by one to obtain the multiple alignment, okay. So we will see in a moment. So progressive methods are Cluster W and muscle. So let's consider an example of two data sets. One is 5 distantly related globins, human to plant, and 5 closely related beta globins. And you can use cluster W or cluster omega to perform multiple sequence alignment. You have to just input the sequences. Each sequences should have identifier line also. Identifier line is greater than, and then name of the sequence, any additional information about the sequence, so description of the sequence. Okay, so and then you can just copy paste, and then you can sum it, and it will provide you the multiple alignment. Okay, so let's discuss the progressive method, the Feng-Doolittle multiple alignment method, which occurs in 3 stages. The first one is that you have to perform global pairwise alignment. So given n sequences, obtain all pairwise alignments, okay. So you can use Needleman-Wunsch algorithm for performing pairwise alignment, so global alignment. And then after you have done the alignment, you can make guide tree, and after you have got the guide tree, you can use that to align the sequences, okay, progressively align the sequences. So let's consider 5 globin sequences: beta globin, myoglobin, neuroglobin, soybean globin, and rice globin, okay. So there are 5 globins, and when you consider all pairs, how many pairs would be? 5 globins, and you have to get all pairs. So how many total pairs? How many? 5 combination 2. 10, yeah, 5 combination to 10. Okay, so 10. So you can see here these 10 pairs. So beta globin aligned to myoglobin, score is 25. Beta globin aligned to neuroglobin, score is 15, and so on. Which has the best score? Soybean globin and rice globin, a score of 43, right? So this is similarity score. But when you make a tree like a guide tree, which is similar to phylogenetic tree, you would want to visualize the sequences in the tree, sequences or organisms or objects, whatever you want to make the tree for. You want to visualize their relationship in terms of distances between them. if you have constructed a tree, then you would want to convert the similarity scores into distance scores. Okay. So that's the next step. So convert similarity scores to distance scores, and once you are done, then you can make a guide tree. So here is a formula actually that can help in converting similarity scores into distance scores. So S observed is the similarity score as you are doing with the blast. Similarity score between two sequences. Okay, that is S observed. S random is the similarity score for alignment of two random sequences. S max is the maximum similarity score, and when similarity score will be maximum? When the sequences are identical. Okay, identical. So you got all this. So so let's consider identical sequences. Okay, so then identical, you have identical sequences. So S observed would be S max, right? So S max minus S rand divided by S max minus S rand, this would be 1. So for identical sequences, distance would be minus log 1, and log 1 is 0. Okay, so distance between identical sequences would be 0, makes sense, right? Distance would be 0. And now let's consider that you have two random sequences that you have aligned. So S observed would be S rand, right? S rand is the score for the random alignment. So S observed would be S rand. So this would be S rand minus S rand 0. Okay. So log 0. What is log 0? No, log 1 is 0. What is log 0? Yeah. Yeah, think what is log 0? So what raised to power of base say 10 will give you 0? Okay, minus infinity. Okay, minus, do it. You will be able to figure this out. So minus infinity. So minus infinity multiplied by minus would be infinity. So if there are two random sequences aligned, the distance between them would be maximum, and here you have converted that distance to infinity. Okay, so this is just a conversion from similarity scores to distance scores. So for identical sequences, distance is 0. For two random sequences, you would imagine that they should have the highest distance. The distance is infinity, right? All else lie between them. So once this is done, then trees can be constructed, and you can see here the tree for the 5 globins. Beta globin, myoglobin, neuroglobin, soybean globin, and rice globin. So soybean globin and rice globin, they have the highest similarity score. So they have the lowest distance between them. So they should be placed next to each other in the tree. So this is shown here. They are placed next to each other. Soybean next to rice, and then next neuroglobin, next myoglobin, and then beta globin. So this is a guide tree. Okay. So guide tree for 5 closely related globins. So these are closely related. So you can see that the branch lengths are smaller compared to this. These are distantly related. Okay. These are highly divergent. Okay. So next step number 3 in the progressive alignment. So once you have constructed guide tree, then you have to perform multiple alignment using the guide tree. So you will start with 2 most closely related sequences. Okay. So multiple alignment you have to do. You have the guide tree, and from the guide tree you know that which are most closely related sequences, and you will put them next to each other when you are constructing the multiple alignment, and you know how they are aligned. Okay. Then you will bring the next sequence, and then next sequence by looking at the guide tree. Okay, in the same sequence as you constructed the guide tree. Okay, So here is the approach. So sequence to sequence alignment, that's pairwise dynamic programming algorithm will help you do that. Okay. So yeah, sorry, it is just like this actually. This course, you have to sit for many hours, 5 hours and 6 hours, and instructor also has to give lecture for many hours. I understand it is not easy for you and maybe challenging for me also. But we all have to bear, and we have to make through. Okay. Okay, so sequence to sequence alignment, but then okay, two sequences that are closely related, you can see that from the guide tree. So this is how you constructed the guide tree. So you will align them first, and you know how two sequences are aligned, right? Then you will bring the next sequence, but then the next sequence has to be aligned with the alignment of the two most closely related sequences, right? So a sequence has to be aligned with a group of two sequences which are already aligned, right? So how to align them? So the approach is, so that is sequence to group. So the approach is shown here. So you have a group of two sequences that are aligned, and then you bring a third sequence and you want to align this sequence against a group of these two sequences, right? How to align them? So to do this, again pairwise approach, you will compare A with B and A with C. So across these two cluster comparisons, one cluster has one sequence, the other cluster has two sequences. So across cluster comparisons, and then you identify the pairs that are closest. Okay. So let's see let's say that when you did this, then so the comparisons are A to B, A to C. Among these, let's say that A is closer to C than to B. Okay. So A is closer to C, so I will align A with C. Okay. So I already did it when I was comparing across the cluster. Okay. So A is closer to C, so I will have A aligned with C. And I know how B aligns with C, right? This was already done. So I will bring B and will align it as was originally aligned. So I know which residues are aligned between these two. Okay. So I will align accordingly, but I may have to insert gaps to align them because they were A and C were aligned. So the only thing that you have to do to make sure that these residues are aligned in the same order as they were originally aligned. Insert gaps to make this happen. Okay. Okay. So which one? A can come. So A and C, yeah. Yeah, you can put A here and B, it doesn't matter. Doesn't matter. Or it is also possible that A and B is closer to C and A and C, right? It may also happen. So in this example, A and C, they are closer than A and B. Okay. So then A and C aligned, and then you bring B here, and you know how B is aligned to C. Okay. 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 And similarly, if there are two groups, so if groups have to be aligned, then again perform a cross cluster comparisons and identify the pair that is the closest. So A compared with C, A compared with D, B compared with C, B compared with D, and identify the pair which is having the closest sequences. Okay. And let's say that the closest sequences are in the pair A to D. Okay. So among these four, A and D, this pair has the closest sequences. Okay. So then A and D aligned. So A aligned to D, and we know how B is aligned to A. So I will bring B and will align to A. making sure that they are being aligned as originally aligned. Okay, so residue should be aligned in that order. Insert gaps as needed, and then you will bring C aligning with D as originally aligned. Okay, because these are frozen, we know how they are aligned, so we have to make sure that when you bring, they should be aligned as they were originally. Okay, so this way, using the guide tree, you are bringing the sequences and you are aligning them one by one in the order of decreasing similarity. Okay, so this is how the sequences will get aligned one by one, guided by the guide tree. So one of the challenges actually, and people think that okay, let's do this, these are heuristics, let's do this way, and we will be able to solve the problem. But you may be wondering, and as you told me, that multiple sequence alignment is more powerful than pairwise alignment because it can capture patterns that pairwise cannot. But here we are using the tricks from pairwise alignment to construct multiple alignment, right? So you would want to just do away with pairwise if possible when you are constructing multiple alignment. So people thought over this and they suggested that let's try another approach where you don't have to be guided by pairwise alignment, because again these are heuristics. So you perform multiple alignment guided by pairwise alignments. Okay, so you would want to just get away from using pairwise alignment to build multiple alignment, because the whole idea of multiple alignment is to address the limitations of the pairwise alignment. So the approach is that if you have the multiple alignments that you want to align against each other, so groups of alignments that you want to align against, then you would want to consider, okay, this is better. You would want to consider the approach that we already discussed. So let's consider, so pairwise alignment is very clear, right? When we begin with pairwise, and then once we build further alignment, so we may encounter a group of one sequence being aligned with a group of two sequences, so group of multiple sequences, then group of sequences to be aligned with group of sequences, right? So so let's consider that okay, we have a group of sequences that have to be aligned with another group of sequences. Okay, and these have already been aligned, so their scores are known. It's frozen, it's already aligned, so frozen scores are known, but we want to align this group with this group. Okay, so we would consider, as we were discussing about optimal alignment of two sequences, we would want to consider all possible lineups of these two clusters. Okay, and then score each of the lineups of the two clusters. So let's consider this lineup and consider all these columns. Okay, so lined up, and then we'll consider the columns. Okay, so okay, this is one column, and then when you score this column, you have to consider all possible pairs of the residues that are aligned in this column, right? This residue aligned with this, what is the score for this? This residue aligned with this, the score for this, and you have to sum them up. But the scores for this aligned to this aligned to this has also already been taken into account when the multiple alignment for this was obtained, right? So and again, this aligned to this aligned to this aligned to this to this to this, and so on. So these within cluster groupings of residue pairs, they have already been accounted for. Okay, So what is new when you compare this cluster with this cluster? Okay, so this aligned with this aligned with this aligned with this with this with this, and this with this. Okay. And then next one is this aligned to this, so a score for this, and then this with this, all possible pairs. Okay. So, but some of the pairs have already been accounted for in the scoring when these two groups were formed. Okay. But what is new here? Was this to this accounted for previously? No. And these are called cross terms. Okay. So this aligned to this aligned to this. So those that are cross, that are a cross between the two clusters, they are the new terms. Okay. So you would want to get the scores for the cross terms. The scores for within cluster pairwise terms, they are already accomplished, they are already done, they are frozen. Okay. So what you have to get is the pairs or score terms that are across the clusters. Clear? So when you are performing the lineup, so for each lineup you have to get the cross terms across the clusters, and you have to maximize it. Which lineup will give you the highest value for the sum of the cross terms? Okay. So this is actually indicated here, the three terms, the cross. So this is frozen, this is for first cluster, this is for second cluster. So this to this, this to this, this to this. Okay. This is the first one. This is already done, right? This is aligned. Then the second one is this to this, this to this, and this to this. This is already done. This is frozen, already completed. Okay. So that is the second one. So they are constant. They are already done. The third one is the new one, which is the cross term. This compared with this across the clusters. This compared with this one with this, and so on. Okay. That is the third one. That is the new. And if you want to maximize the score, you have to maximize the third term, the cross term, right? And identify the lineup of two profiles or two groups of sequences that has the highest value for the third term, right? Because this is constant, this is constant, this is what is changing. You want to maximize this. Which lineup of the two clusters of aligned sequences yields you the maximum value of this third term, the cross term. Okay. So this is what results in profile alignment. So this is how they could get away. So developed by researchers who worked on multiple sequence alignment, they proposed that using the profile alignment is more robust compared to the pairwise alignment for these regions. Okay. Okay. So now we can go through the steps of cluster W, which is one of the most used programs for multiple alignment. So you will construct a distance matrix for all pairs by pairwise alignment. So you will get the similarity scores for all pairs, and then you will convert the similarity scores to distance scores. Okay. And then you will make the guide tree. You will make the guide tree and then use the guide tree to progressively align the sequences, beginning with the most closely related sequences. So sequence to sequence alignment, and then sequence to group, and then group to group alignment using the profile alignment approach. Okay. In the order of the decreasing distance, beginning with the closest, and then as you iteratively align, you go to the more distant ones. Okay. And at the end the most distance. So all those then sequences will get aligned this way. Okay, so here is an example of 5 distantly related globeins. So you can see terminal gaps and internal gaps. There are few positions which are fully conserved and they are indicated by stars, very few. And there are some positions which are conserved, they are shown by dots or columns. And here is an example of 5 closely related globeins, and you can see a very good conservation. There are many positions with complete conservation and I don't see any gaps. So, and these are beta globeins from human, chimpanzee, dog, mouse and chicken. Okay, highly conserved, you can see this. Okay, so there could be a problem with progressive alignment, and this is something that we were already discussing, that once you have the profiles from earlier stages of multiple alignment, it is frozen. You don't change it. Scores are frozen, alignments are frozen. But it is possible that at the earlier stages there might have been some errors in the alignment. But because you keep it frozen, it is never changed. Even at later stages of the alignment, you may find that there are some problems that happened in the earlier stages. This method is not going to change. Okay, so which means that this is a heuristic method, which is a rigid method, sorry, a rigid framework that does not allow change even though mistakes might have happened at earlier stages of alignment. Okay, so then people thought about approaches that can help in defreezing this alignment and increasing the score of the alignment, which means that making it better. And one of the approach is called iterative refinement. Okay, iterative refinement means that once you have generated multiple alignment, then take out randomly one sequence from the alignment and align that sequence against a group of the remaining sequences using profile alignment. You learned about profile alignment. So you have a group of one sequence and a group of another sequences, and then you perform alignment of those two using the profile alignment. Okay, so the main idea here is that when you will perform that, either the score will increase or a score will remain the same. Okay, so you do this, as you will do this, you should see that a score is increasing and go through many iterations until convergence. Convergence means that if you do it again, the score is not going to change. Okay, so this is called iterative refinement. And here is a method that uses iterative refinement, and this is called Barton-Sternberg multiple alignment algorithm. So the first step is find two most similar sequences using dynamic programming algorithm, Needleman-Wunsch algorithm, I think global alignment algorithm, and put them next to each other. Okay, so most similar sequences you will put next to each other and bring the next sequence which is closest to the group of these two. Okay, so then you will have group of 3, right? And then bring the next which is closest to this group of 3 now. So then you will have group of 4, and then bring next until you have aligned all the sequences. So another approach, heuristic approach. Okay, so once you have aligned, what you will do, you remove the first sequence and then align it against the group of the remaining sequences using the profile alignment. Okay, then remove second sequence and align it against group of the remaining sequences. Okay, repeat it iteratively. Third sequence removed, aligned against group of remaining sequences. Then fourth sequence removed, aligned against group of remaining sequences, and so on. Do this process several times some defined number of iterations or until convergence. Convergence means that a score is no longer increasing. Okay, done. Yes.
So in profile alignment, we can align the middle sequences also with the across the group, or is that only after iterative?
So you align the group with the group.
Yeah.
Right. So, so not any particular middle or any particular sequence, you align the whole, because this is frozen. You cannot change it. So, you align this group with this. So, this is also frozen. Okay. So, then for example, this is the best one. So, you can see the alignment that these residues are aligned against gaps, right. So, these are aligned here and then there are gaps here.
Okay, so the whole group at once.
Right, right. So then this is a multi. So think like that. If this is the best, then then what should I say that? Okay, this is the first column. Okay, so then there are gaps here, gaps, gaps, gaps. So okay. So we'll write actually in the same order. So gap, 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 gap. Okay. So this is the first column. Let's say that this is the first column. Second column. So this, and then again there is nothing. So gap, 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 gap. So this is and so on, right? Clear? So yeah, you have these columns until you hit here, and then you have residues from this group aligned with residues of this. Okay, so when you come here, then you have this column where you have residues aligned against each other in this group, but here there are gaps. Okay, so this is how okay, it's fine. This is how the two groups are getting aligned. Okay. Okay. So these programs use iterative refinement methods. They map, interalign, proline, and muscle. Okay. Okay. So when you have constructed multiple alignment, you can use that for making phylogenetic tree. Because okay, so maybe couple of minutes and then we will resume tomorrow. Okay, it's 4:33. So multiple alignment is one of the requisite of phylogenetic tree construction. So first you have to identify homologs and then perform multiple alignment of homologs. And once you have the multiple alignment, then you can use methods for tree construction that can help make a tree of these homologs that have been aligned using multiple alignment method. So we will develop how to construct a tree once sequences have been aligned. Okay, tomorrow.